we come to our next lecture on carbohydrates, and here we will be looking at the formation of disaccharides and the formation of polysaccharides and how they actually help, or where they are found and what linkages they actually have. So, before we get into that, we will look at what these carbohydrates are. We learnt in the last class that carbohydrates have the general formula CH2O whole N. Okay. Now, this is something you have studied from probably your school, where you know what starch is and what cellulose is and things like that. But what we are going to look at now is what are the specific monosaccharides that form starch, what are the linkages that result in the formation of starch and in the formation of the other polysaccharides, and why, for example, there will be certain enzymes that do not work for us humans, but work for animals. So, we have starch, that is nothing but a polymeric form of glucose found in plants, and glycogen is a polymeric form of glucose that is found in animals. Okay. So, since you understand now that since the basic unit is glucose, it would be the linking that would be different, because now that is exactly what we are going to consider, is how these glucose units, these monosaccharide units are linked with one another. And these are both immobilized. Glycogen is stored in our liver and it is broken down when we require the glucose breakdown for metabolism for the energy requirement of our activities. Cellulose is another major polysaccharide in plants that serves rather to be a structural than a nutritional role. Okay. Structural means it forms fibers that help in its structural build-up. Now, what we have here is certain types of bonds that we are actually going to be looking at. Glycosidic bonds are those bonds that link sugars to one another. Okay. Now, when we consider, say, just glucose linking with another glucose moiety, or glucose linking with galactose or mannose, or where we have just O glycosidic linkages, we are going to form disaccharides and polysaccharides. So, the O glycosidic bonds that link sugars to one another in disaccharides and polysaccharides. When we speak of N glycosidic bonds, we know that in the O glycosidic bonds we have the oxygen that is involved in the linkage. When we have an N glycosidic bond, we know that the nitrogen is involved in the linkage, and when you have glycosidic, it means that there is a sugar involved. Okay. So, we have nitrogen and sugar. These are the bonds that link sugars to the purines and pyrimidines in nucleotides. This we will study in much more detail when we consider the nucleic acids and their specific components. But for now, what we studied in the last class, when we look at ribonucleic acid, it has a ribose sugar to it. Now, to the ribose sugar, we know that the ribose sugar itself contained only carbon, hydrogen and oxygen in the carbohydrate unit that it was. Now, if we have the purines and the pyrimidines that are actually nitrogenous bases linked to the ribose or the deoxyribose, there is a specific N glycosidic bond linkage that comes into play. So, when we have an O glycosidic bond, we are basically forming a disaccharide, because we have two monosaccharide units linked by an O glycosidic bond that is going to form a disaccharide. And similarly, we will have another set of linkages that are going to result in the formation of polysaccharides. In case of N glycosidic bonds, we are going to have this linking of the sugars, the ribose or the deoxyribose to the bases, purines and the pyrimidines. Another important part is phosphorylated sugars. What does that mean? It means that when you have a ribose sugar, you can have phosphate attached to it, which we will see when we consider adenosine triphosphate. where we have phosphorylated sugars that are key intermediates in energy generation and biosynthesis. We have the disaccharides that consists of 2 monosaccharide units, 2 sugar units that are joined by an O glycosidic bonds and glycoside bond, and the 3 most abundant disaccharides are sucrose, lactose and maltose. Okay, these are the ones, but we will know how, once we know what the linkage is, how we can actually write out the structures of definitely sucrose, lactose and maltose, but also of other disaccharides. Now, this is something that I was mentioning in the last class. We are concerned here by the type of glycosidic linkage we have. Okay? Now, in forming a glycosidic linkage, we have a linkage depending on what the first monosaccharide has a disposition of the C1 carbon atom. For example, if the linkage is between a C1 alpha OH and a C4 OH, it does not matter what the orientation of this C4 OH is. If the first carbon is an alpha, it is called an alpha glycosidic bond. If it happens to be a beta, it is the linkage between a C1 beta OH and a C4 OH. It is called a beta glycosidic bond. Let us just go here and just look at how we had hemiacetal formation. Okay, this is something that we did in the last class. This was our aldehyde molecule. When we had the aldehyde molecule, we know that it cyclized. Now, when it cyclized, we had the C double bond O, that is the aldehyde part, the aldehyde moiety, interact in the case of glucose with carbon atom number 5, the OH of carbon atom number 5, into forming a cyclic structure, which is the hemiacetal. Now, what happens in this case? You still have an OH available to it, right? And now what can happen is we can have another unit come and link up with the hemiacetal into forming an acetal. Now, this unit which has an OH group could be another sugar, right? It could be another sugar also. So, we are going to form an acetal. In the intramolecular cyclization of the glucose forming the cyclized monosaccharide, what was actually formed was an hemiacetal, right? Because we had the OH that actually was part of R1. So, this R2 that we are talking about here is actually the 5th carbon atom of the same structure here. So, we are cyclizing this whole thing into forming what is called a hemiacetal. But now, if I have another glucose which is also an OH, it has an OH to it, right? If I have this link with this hemiacetal, what can I get? I can get an acetal. Now, depending on my cyclic structure, I have what? I have either an alpha form or a beta form, right? Let us just draw one form here, or in fact, let us draw both forms so we know what we are talking about. We have two forms here. What are these? These are pyranose rings. We have our CH, which carbon atom is this? It is the 6th carbon atom. We have our OH here, one OH here, one OH here, and this OH we know can be either up or down, right? If I put it down, it becomes alpha. If I put it up, it is beta. Okay. So, I am not drawing the other one. So, this is our beta, this is our alpha. Okay. Now, if I form a linkage, then the oxygen atom which is going to be linked. Now, suppose we have these two units. If it is in the alpha form, the oxygen is where? It is down. So, the linkage is going to look like this. The oxygen is going to be down. It can be down. But the other one that it's linking with can be up or down. So, this could either be this or it could be this. Is that clear? What we are looking at is we are not looking at our monosaccharide unit only anymore. We are going to link this monosaccharide unit with another monosaccharide unit into forming a glycosidic linkage. In the formation of the glycosidic linkage, we have to look at the disposition of the OH with respect to the CH2OH that is going to determine whether this is alpha or beta. Okay? Now, these are both alpha forms. Why? Because in both cases my OH is down. This O that is coming from the first monosaccharide unit is trans with respect to the CH2OH. That is why the direction is this way. If I were to do the same for the beta set, then in both cases it would be up. What are both the cases I am talking about? We would have an O and an O, and the other monosaccharide unit that it was be linking to could either be this or this. Is that clear? Because what we are looking at now is we are going to gradually get onto structural units. Now, to understand structural units, we have to be very careful about the linkages we are talking about. Because if you remember when we did lysozyme, we talked about beta 1,4 glycosidic linkages. What are these linkages? A beta linkage would look like this. Okay? Now, in both cases, it would begin up. Why? Because this is up. This is cis to the CH2OH. So, this would refer to a beta linkage. In this case, it is down. It is trans to the CH2OH. So, this would refer to an alpha linkage. And what it is linking to could be up or down. Right? So, we have this either looking like this or like this, and the same thing in the beta case. Okay? That is why we have suppose we have it linking to the C4, which is the fourth carbon of the next monosaccharide unit, then the C4 end could have the oxygen where? Up or down, depending on what unit you are considering. Now, if that is the case, then you would have, dependent on the orientation of the monosaccharide, either it come down. In the alpha case, it always has to come down. That is what you have to remember. Why? Because the oxygen, or the OH rather, is trans to the CH2OH, and it is this that is going to be involved in the acetal formation. We already have a hemiacetal formation. We are now going to have an acetal formation. So, what is happening is this OH is going to be involved in a reaction that is going to form a glycosidic linkage. Now, when it forms this glycosidic linkage, in the case of the alpha, both of them are going to be down, and the one that it's linking to, say the C4 and in this case, can be up or down. The same goes with the beta. The beta will be up initially. Why? Because the OH is cis to the CH2OH, and what it's linking to could be either up or down. Okay? So, this is what we have. We can have alpha bonds that look like this. You have to remember, when the O looks like this is an alpha linkage. When the O comes from the towards the top or upward, it is a beta bond. So, this is what it looks like. So, depending on what kind of linkage you have, you see how the structural differences occur. That is obvious. Okay, it is going to be obviously dependent on how the reaction is taking place. Okay, so you could either have an alpha glycosidic linkage or you could have a beta glycosidic linkage. So let's see what it is. So this is what we have. We have an alpha D glucose here and a beta D glucose. Okay, now follow this very carefully. This is the structure that we did in our last class. What do we have? We have hemiacetal formation where the OH that is of this carbon, what carbon is this? Carbon number 5. And this is carbon number 1 that originally was an aldehyde. It is now a hemiacetal due to an intramolecular cyclization that has occurred between the OH at the 5th carbon and the aldehyde. The OH is trans to the CH2OH. So this is alpha D glucose. The one on the right here is beta D glucose. Why? Because the OH is cis to the CH2OH. This is a hemiacetal. It still can react with what I showed in the first diagram here, where we have an R3OH. So what is our R3OH? This is our R3OH, right? So where is the, which OH is this? This is the OH on the 4th carbon atom of another glucose moiety, another monosaccharide. So what can it then form? It can form an acetal, right? When it, so initially we had the straight chain which actually linked these two units up together to form an hemiacetal. Once the hemiacetal was formed, this is now linking with another monosaccharide unit to form an acetal. Okay, so that's exactly what is happening here. We have alpha D glucose, the hemiacetal, which is linking with what number carbon is this? Carbon number 4. This is carbon number 1, 2, 3, 4. Let me just get this straight here. So what we have is, what is this carbon number? 1. This is carbon number 1, this is carbon number 2, this is carbon number 3, this is carbon number 4, this is carbon number 5, and that's carbon number 6, right? So what is the linking? We are linking this, which is a hemiacetal, with the OH here. So we have HOR3. What is R3? R3 is the rest of the molecule. So this is our hemiacetal now, that is going to link with this alcohol to form an alpha 1,4 linkage. Why alpha? Because this is alpha. So when what is it going to look like? It is going to what is it going to look like in terms? It is going to look like this. It has to come down. Why? Because it's alpha. And depending on the OH orientation of the C4 carbon, this is going to be up or down. In this case it is up. Is that clear? So what we have is, we had our glucose. It has cyclized to form the hemiacetal. We now are linking it with another monosaccharide moiety to form a disaccharide. Okay? The linkage is a glycosidic linkage because we are linking sugars. It is an O glycosidic linkage because the linkage is between the sugars by an oxygen. The linkage to the sugar is by the oxygen. So this is an O glycosidic linkage. We are linking the alpha of carbon number 1 to the 4th carbon of the other one. It does not matter whether this is an alpha or a beta. The alpha comes from what is the first one that you are considering. This is alpha, carbon number 1. So, this linkage is an alpha 1 4 linkage. It is linking what? It is linking, this is the actual nomenclature, where you have alpha D glucopyranosyl 1,4-D glucopyranose. What has it now formed? It has formed an acetal. This is the R3 moiety, remember? This is our R3 part, this is the OH part. It has now formed an acetal. Okay? And what can, this is now a hemiacetal. What can happen? This can link with another sugar, right? So, we can have a polysaccharide. So, this is the formation of a disaccharide, and we are going to look at specific linkages, because if this name is given to you, you should be able to draw the structure of the disaccharide formed, because you know that you have, the structure of glucose. You know that if you have 2 glucopyranose units, it means you have 2 glucose units, and if the linkage is alpha 1,4, it forms maltose. Okay? So, we have alpha D glucose linked with beta D glucose. In this case, it is beta Y because it is a cis, but that has nothing to do with the linkage that we are concerned with here. When this is going to react with the next R3OH or next ROH, what is the linkage going to be? It is going to be a beta linkage, because the OH in this case, where is our OH? The OH in this case of the hemiacetal is up. So, the linkage is going to be something like this, and we do not know what is going to be the other one. So, it could be either up or down, but this is beta, this is alpha. Is that clear? This is about the glycosidic linkages in the formation of disaccharides. Okay. Let us look at a few more. Okay. So, what do we have? We have this going up. So, what is this? Beta. It is linking with the 4th carbon. Is this the 4th carbon? This is O, this is 1, 2, 3, 4, 5, 6. So, what linkage is this? It is a beta 1, 1, right? So, we have to be careful at what we link, because now you formed a completely different disaccharide. Okay? So, because you have to know that the numbering, this CH2OH is number 6. So, 6, 5, 4, 3, 2, 1. Okay? So, we have, and this is number 1, it is going up, so it is beta. So, it is a beta 1, 1, O. Why O glycosidic linkage? Because we have the oxygen bridge there. That is exactly what is happening in the glycoside formation. Okay? Now, let us look at some examples. So, okay, let us look at the rules first. If the alcohol linked to an anomeric carbon is another carbohydrate, obviously the other resulting compound is a disaccharide, because we have 2 monosaccharide units. The name of the glycoside specifies the 2 monosaccharides and the glycosidic linkage, which carbons are linked, and the configuration about the anomeric carbon, the alpha or the beta. Okay? So, we have either the alpha or the beta, and we have to figure out what it is linking to, or which carbon it is linking to. Now, these are some examples. Lactose is galactose beta 1,4 glucose. So, what do we need to know for this? We need to know the structure of galactose, we need to know the beta anomer of galactose, and the structure of glucose, and then this is the way it is written in short form, galactose beta 1,4 glucose, that is lactose. If we look at sucrose, it is glucose alpha 1,2 beta fructose. All of you know what sucrose is, so you can check and see whether this is the right way to write it. So, we have glucose alpha 1,2 beta fructose. If we looked at trehalose, it is glucose alpha 1,1 alpha glucose. Okay. Maltose is the one that I showed you previously, where we have a glucose alpha 1,4 linkage. That is what we showed when we were looking at the acetal and the hemiacetal. We have cellobiose. Okay. Let us do just one of these structures. Say let us do trehalose, okay, because it is both glucose units and we already looked at maltose. So, we are looking at, what is this? This is glucose alpha 1,1. Actually, alpha 1,1 alpha glucose is written here, which means that the other glucose is also alpha. So, this is what we have when I write this. How will I write the O? Down, right? Okay. Now, what happens to the other one? How do I write this? Now, I have it linked to number 1. So, my oxygen has to be, where does my oxygen have to be? It has to be here, right? And where is my CH2OH? It is here. It has to be linked like that. It's actually, the link is like this. Okay, I couldn't draw it correctly here, but that is what your structure is going to look like, where the H is going to be here. It's basically going to be like that. Okay. So, we have, what do we have here? What does this signify? Alpha. It is carbon number 1. This is carbon number 1. So, 1. These two units are glucose and glucose. So, glucose alpha, should be alpha 1,1. That gives rise to trehalose. If we look at, then look at all the different monosaccharide or disaccharides rather that we have here. We have lactose, sucrose, trehalose, maltose, cellobiose. Okay. This is lactose then. What is the linkage? Just let us look at linkages now. Beta, try not to look at the answer, but just to figure out that we are linking, this is the 6th carbon, this is the 5th carbon, that is the 4th one. So, beta 1,4 linkage. Okay. It is linking galactose and glucose, that is lactose. Okay. So, it is the dimer of beta D galactose and either alpha or beta D glucose. So, this could be either up or down, this OH, because it is linking either the alpha or the beta D glucose. So, we have beta 1,4. In cellobiose, we have, this is also beta 1,4. What is this? Alpha 1,4. This is maltose. This is the first example that I showed you. Now, just because this linkage is different, we have an enzyme that can break this disaccharide down, but we do not have an enzyme that can break this disaccharide down. Okay? It is that specific. All the enzymes that break down sugars, which we will see later on, we have an enzyme that can break maltose, but not an enzyme that can break cellobiose, and the only difference is that this is a beta 1,4 linkage and this is an alpha 1,4 linkage. Okay? This is actually what it would look like if you were to actually draw it correctly. This would be alpha 1,4 linked D-glucose units. So, what can we have? We can have other units linked together. So, the whole structural aspect comes into the picture now, as to, because we are actually not going to have those projections that we are drawing, but we are going to have actual chair conformations. Now, when we have the chair conformations and we know that we have equatorial and axial bonds, then obviously we are going to have the linkages that are going to result in different structural aspects to the whole disaccharides and polysaccharides. Okay? That is why each of them are going to behave in a different structural fashion. Okay? So, depending on the linkage, it could be starch, it could be glycogen, it could be amylose, it could be amylopectin, whatever. Okay? And that is what we are going to look at. The polysaccharides. The polysaccharides therefore will be further linkages of what we just learnt. Okay? So, not only will we have an alpha 1,4, we could have alpha 1,4, alpha 1,4, alpha 1,4, beta 1,4, so on and so forth. These polysaccharides are just therefore polymers of the monosaccharides, and they could be storage type. The storage type are starch and glycogen. They could be structural, where they are used to provide protective walls or lubricative coating to the cells. This is like cellulose and mucopolysaccharides. We will see an example of each of these as we go along. You also have structural peptidoglycans that actually form bacterial cell walls. Okay? And what did we learn when we did the lysozyme structure? That it breaks bacterial cell walls. Okay? So, we can have polysaccharides that look like this. So, these would be just normal linkages, 6-membered rings. You could have homopolysaccharides where all the sugars are the same. You could have them unbranched or you could have them branched. So, you would have say an alpha 1,4 linkage here and say a 1,6 linkage here, or a 2,6 linkage there, which would form a branch to it, right? You could have heteropolysaccharides where the monomer types are different. You could also have them unbranched or branched. So, these would basically be the 4 types of polysaccharides that you could actually have: 2 types of homopolysaccharides and 2 types of heteropolysaccharides, meaning they could be unbranched or branched. Okay? And each of the units, the monomer units that we studied in the last class and this class in the disaccharide formation, will account for all types of polysaccharides possible. Okay? So, what are these types? Starch. Starch and glycogen are both alpha 1,4 linked glucose polymers. You now know what that means. Alpha 1,4 linked glucose polymers. So, it is glucose, alpha 1,4 and another glucose that are periodically branched with alpha 1,6 linkages. So, you have alpha 1,4, you can also have alpha 1,6 that is going to basically make it a branched homopolysaccharide, right, because you have the same monosaccharide unit. Glycogen is highly branched. Every 8 to 10 glucoses with average side chains of 8 to 12 glucoses. Means you have in your previous picture, if you look, you have 8 to 10 here and 8 to 12 branched off, then another set. So, you have all of it linked together basically to form, and you realize now that it is good to form such a compact unit. Why? Because they are storage polysaccharides. You do not want them to take a lot of space. Okay? So, you would rather want the storage polysaccharides to be compact, which is why starch actually forms granules. Okay? For storage purposes, it is more convenient to have these. Okay, now starch, we will look at the structures in a bit more detail. Starch consists of amylose, that is completely unbranched, and amylopectin, branched once every 12 to 25 glucoses. So, an unbranched polysaccharide, a homopolysaccharide. Why? Because it has the same units. An example of an unbranched homopolysaccharide is amylose. Okay? And these are examples of branched ones. Then the alpha 1,4 linked glucose polymer units in amylopectin actually form something that looks like a helix, because they link, one linked like this, and like this. I will show you in a picture right now. In cellulose, you have a beta 1,4 linked glucose polymer. Okay? That is used by plant cells. Unlike the storage forms which I was mentioning, cellulose is not branched, because it would not be useful to have something fibrous that is going to be a structural polysaccharide to be compact. It should be fibrous. So, if it is fibrous, it has to be a long chain. It cannot form a unit. Then the neighboring chains are linked together by hydrogen bonds. Okay? So, then we have other examples which I am not going to go into, but basically we have chitin, we have hyaluronic acid, which actually have linkages of that N-acetyl glucosamine. If you remember, we did N-acetyl glucosamine and N-acetyl muramate, the NAG and the NAM, when we considered the lysozyme cleavage, right, how it could actually cleave. So, this is basically where it is going to work. So, what we have here, this is what it actually looks like. We have, what sort of a linkage is this? Beta 1,4. This is an N-glycosidic. This is the CH2OH, which is 6. Right? If you get confused with where the 1 begins from, go to the CH2OH. This is 6, 5, 4. So, a 1,4 linkage. This is what? It's an N-acetyl. Right? This is an example of an N-acetyl. You look at the ends, what are they? So, what is this? This is actually a glucosamine. The OH, there is no OH there. What is it? It was an NH2 that has been acetylated. So, it is a glucosamine. Okay? And this is a beta 1,4 linkage of a glucosamine. So, what is it? Here, you have beta N-acetyl glucosamine. So, you now know what it looks like. It is a glucose which had an OH changed to an amine which is acetylated, that is linked by a beta linkage. Okay? So, this is basically what we have. What is this now? Alpha 1,4, right? Alpha 1,4, one after the other, then suddenly you have some 1,6. So, you see how it looks like a circle like that. So, you have like a helix structure. Okay? This is amylose starch. What you need to know is how you can actually form these linkages, an alpha 1,4 linkage. So, if you know that the straight chain forms alpha 1,4 linkages that then branch off to form 1,6, it looks like that. If you look at amylopectin, you have, what is this? What is this linkage? A beta 1,4, that at the cross link, where is the cross link? Here. No, that is not the cross link. Where is the cross link? The alpha 1,6 linkage is supposed to be at the cross link. So, where will be my cross link? It will be this. This is the 6, right? That is the 6th one. Okay, so these are basically the different types. Here again, you have glycogen that looks like this, and what we have to remember is when you are looking at storage polysaccharides, they are stored as granules. That is why they have to be branched polysaccharides. Okay? That is an important feature of branched polysaccharides. Again we have cellulose. Cellulose does not have to be branched. In fact, it should not be branched. It should be long fibrous for the plant structure. So, this is basically the most abundant polysaccharide, and this is a beta 1,4, right. Then we have mucopolysaccharide. What do we have? What is this unit now? The NHCOCH3, N-acetyl glucosamine. This is glucuronic acid. What do we mean by glucuronic acid? This is not CH2OH anymore. It is COO minus. Okay. So, we have N-acetyl glucosamine and glucuronic acid, which is what formed in the Felling's reaction also. You have the COO minus instead of the CH2OH at position number 6. This is probably something we should be looking at, where we have peptide crosslinks between the polymer strands. Now, the reason we have peptide crosslinks is, where did we see this? In bacterial cell walls, when I was just mentioning about lysozyme. We had alternating units of N-acetyl glucosamine and N-acetyl muramic acid. Remember, we have the muramate and the glucosamine. So, what we have is, in between these units, where we have an R group, this is the R group. This R group is actually some alanine, lysine. So, it has a peptide unit to it. Now, because of this peptide unit, it imparts structural rigidity to the cell wall. Okay? So, it is nothing but having some peptide crosslinks. Apart from the normal polysaccharide crosslinks, if you have additional peptide crosslinks, there is going to be extra strength to the bacterium, because you do not want it to be broken down. The bacterial obviously does not want its cell wall to be broken down. So, it has extra crosslinks. Now, we have been speaking about glycoproteins when we considered lipids and membranes. Okay? Now, these glycoproteins are what? They are proteins that have linked to them covalently sugar units. Okay? Now, we are going to see where or what these sugar units are and how they are linked to the proteins, because we now know the protein structure, we know the amino acids in proteins, we know the monosaccharides now, right? We know something about the carbohydrates now. So, what we are going to look at is how they are linked together. These glycoproteins have important biological functions. These are some of the functions that are listed here: immunological protection, cell-cell recognition, blood clotting, and host pathogen interactions. Okay? So, usually the carbohydrates account for only 1 to 30% of the total weight of a glycoprotein, and the most common monosaccharides that actually link up are glucose, galactose, mannose, NAG, and NAM. Okay, this is N-acetyl galactosamine, N-acetyl glucosamine. We also have muramate, sialic acid. These are all the monosaccharides that link with the proteins. But where do they link? It means they have to link to specific amino acids. If they are to be formed, if they have to be glycoproteins, it means there has to be a sugar unit that is linked to the protein. So, what is it linked by? If this is our polypeptide chain, there are different ways in which these sugars can actually link. Sometimes you can have, we learnt about O-glycosidic bonds. What are O-glycosidic bonds? Glycosidic bonds that use the oxygen to link, right? Now, in the case of serine and threonine, we know what their side chains are. Serine is OH and threonine is CH3 and OH, right? If we have the linking of the sugar to the proteins, this is the polypeptide chain where we have a threonine. This is the CH3 of the threonine, this is the OH. This OH has linked with a unit, a saccharide unit, This is an N-acetyl glucosamine. And what has it formed? If this is part of the polypeptide chain, as soon as it links up a monosaccharide unit to it, it forms a glycoprotein. And this again you can realize that this can be linked further. So, these were all the carbohydrate units that we had when we looked at glycoproteins in the lipid membrane, the fluid mosaic model for the membrane. When we are forming N-glycosidic bonds, we are using the side chain amide nitrogen. Which two amino acids have amides? Asparagine and glutamine, right? So, now we have to remember that when we are forming a glycoprotein, all we are doing is we are linking the sugar to the protein. Now, this sugar has to be linked to the polypeptide chain through amino acids, because that is what is forming the polypeptide chain, right. So, when we form these glycoproteins, it is the threonine OH that can link to form an O glycosidic linkage, or it is the amide N nitrogen that can form an N glycosidic linkage, fine. Then we can have esterification, which is probably one of the most important biological esters that are formed, where we have a specific enzyme. Remember, I mentioned kinases as being a type of transferase. We have 6 different types of enzymes. Of those enzymes, there was one enzyme called a transferase, one type of enzyme called a transferase. But when we have a kinase, it is a specific type of transferase that transfers phosphate moieties. Okay? We are going to see a lot of these kinases when we do bioenergetics. But for now, we have an ROH. Now, this OH can be part of a monosaccharide unit, right? The OH that we have here can be part of the monosaccharide unit. In that case, we have, what do we have here? We have phosphoric acid. We have the formation of an ester, a phosphate ester and H2O. When we look at a biological system, the formation of a biological ester, then the phosphoryl group from ATP, the structure of which we are going to study in a bit detail when we get to it, the ATP will form an ester with D-glucose into forming glucose 6-phosphate, which is the first step in the breakdown of glucose. Okay? So, we have glucose 6-phosphate and adenosine diphosphate, because it has lost one phosphate to the glucose. Okay? So, this is one sort of reaction we have, and these are the amine derivatives which we mentioned before. What are these amino derivatives? Where this is a beta glucose, you now recognize, because the OH and the CH2OH on the same side. We have beta D glucose, and this is beta D2 amino glucose, which is a glucosamine. It is the OH of carbon number 2 that has been replaced. This is why we have the 2 here, the 2 amino glucose. So, we have the beta D glucose that forms the beta D amino glucosamine, and this can further be acetylated to form NHCOCH3, right. So, these are basically the different types of reactions we can have. Now, the uses for these amino sugars actually are for the structural components of bacterial cell walls, where again we looked at the NAG and the NAM. As a component of chitin, which is a polymer found in the exoskeleton of insects and crustaceans. Okay? So, this is another amino sugar. Now, the formation of the amino sugar actually is important in giving its a structural rigidity. Okay? So, that is why it is sort of a hard shell that is formed. So, basically what happens is we have the NH2 group here, the NH2 that links with an acetyl group, the NHCOCH3 and so on and so forth, forming the different possible chains that we can have. Okay. So, we have different types of oxygen linked and nitrogen linked amino acids, or basically glycoproteins. What are these? When they are linked or when they are O-linked, they are linked through either serine or threonine. What are you linking? You are linking, in this case, if I say GAL-NAC, what does it mean? N-acetyl galactose. Okay? If you know the structure of galactose, the usual N-acetylation is at position 2. We form originally NH2, which is the glucosamine or the galactosamine in this case. If you have galactosamine that is acetylated, you have galactose N-acetylated that is linked to the serine of the peptide chain. So, what has this formed? A glycoprotein, right? So, we can have a serine threonine linked, basically. We can have different functionalities, different types of groups and so on and so forth. In the N-linked case, what do we see? This is again glucose N-acetylated, right? So, it is N-acetylated glucose that is nitrogen linked. So, it means that the amino acid that it is linking to is an asparagine. Okay? Usually it is asparagine and sometimes it can be glutamine. But in this case we are going to have an N-linked, in this case we are going to have an O-linked. Okay? So, in conclusion, what we studied was, we looked at all the monosaccharide units, then we looked at the ways we could form the disaccharides. What are these ways? We have specific alpha and beta linkages, glycosidic linkages that we could use to link the monosaccharide units together. Then obviously, if we can link 2 units, we can link multiple units together. We can have homopolysaccharides, we can have heteropolysaccharides, and they can be branched or unbranched, and depending on how they are utilized or what their actual functions are, they are either cross-linked or not cross-linked. For example, when we have storage polysaccharides like starch and glycogen, they are most likely to be cross-linked. But if we have cellulose, then it is unlikely to be cross-linked, because it would like to be a fiber for structural strength. Okay? And then we later went on to look at glycoproteins to see how the glycogen or the sugar units were actually linked to the proteins. They can be linked to the serine or the threonine via the oxygen, where they are O-linked, and they could either be linked to the asparagine where they are N-linked. Okay? So, that completes our lesson on carbohydrate. We will next do vitamins and coenzymes. Thank you.